This is the final installment of the eye module for the lecture portion, and it's going to focus on the physiology of vision. First thing is a 2-minute video that gives you an overview.
Your eyes allow you to see the world around you, but have you ever wondered how they work? First, it's important to know that vision depends on your brain as much as on your eyes. Your eye's main job is to detect patterns of light. Then, they work with your brain to turn those patterns into images. Let's take a closer look. Light rays bounce off an object you are looking at. Let's say the object is a dog. The light reflects off the dog to your eye. Then, the light enters through the outer part of your eye, called the cornea. The cornea is clear like a window. It helps your eye focus the light to make things look sharp and clear. Next, the light rays pass through an opening called the pupil. The pupil is the dark round circle in the colored part of your eye. The colored part is called the iris. It controls how wide the pupil is and how much light can pass into your eye. In bright light, the iris narrows the pupil, reducing the amount of light that enters the eye. In dim light, the iris widens the pupil to let in more light. All of this happens automatically. Behind the iris is the lens of the eye. It helps focus the light coming into your eye so you can see things clearly. The lens flattens so you can see things that are far away, and bends so you can see things up close. When the lens, cornea, and pupil are all working together properly, they will focus light on the back of the eye. That's important because lining the back of the eye is the retina. There are about 130 million tiny cells in the retina that are sensitive to light. When these cells detect light, they turn it into electrical signals. Those signals eventually make their way through the optic nerve, which is like a cable connecting the retina to the brain. The retina helps create a rough image, but it sees the world upside down. It's your brain that turns what you see right side up. Also, when you look at an object, each eye gets a slightly different view of the world. The brain combines those views and makes them into one picture. The brain also adds a lot of details to your vision, so that you can see complex shapes, movement, depth, and a rainbow of colors. And of course, the brain connects your sense of sight to things you already know, so that when you see a dog, you recognize it as a dog, and not a cat or a monkey. And that's how you're able to see. To learn more about how your eyes work, visit nei.nih.gov/kids.
Okay, so as part of that overview, the pathway of light, you know all of the anatomy of the eye now. And so that light, as it comes in, it is going to go, let's think about the clear structures or spaces that this light would be passing through. First thing is going to be the cornea. The cornea is part of the fibrous tunic. What is that space between the cornea and the iris? The anterior chamber, and it's filled with what substance? The aqueous humor. That's a clear, watery substance, so the light can pass through that. And then they talked about the pupil, the hole in the iris. That light continues through into the posterior chamber, still filled with aqueous humor. And then you've got the lens. The lens is also a clear structure. And that lens is going to flatten or puff out depending on how far that object you're looking at is away from you. And you can see that those light rays are being converged to a point. And the point is going to be something called the fovea centralis in the macula lutea. And that's where you're going to have photoreceptors called rods and cones that absorb the light and convert it into action potentials that go to the brain so that they can be processed by the brain. Note that the object that you're looking at, the light bounces off that object, comes through the clear portions of the front of the eye, and then is going to actually send an image or the signals of the image to your brain that are upside down, because a lens will always invert an image. But the brain is smart enough to realize that it needs to flip it back to upright. The photoreceptors that are in the retina come in two flavors, rods and cones. And in this chart here, it shows that the rods are very sensitive to low light, and it's going to be shades of gray, whereas the cones are for color. Now, what's nice for that, you know, I love my alliteration, but that cones and color both start with the letter C, so that's how I remember those. Rods, however, these are the ones that are going to be used if you walk into a dimly lit room or a dark room that you can kind of make out the shapes of things, but it's kind of fuzzy. Whereas if you have full light, now you've got your cones that are stimulated and it's sending even more information to your brain, and you're getting a much clearer and color image. These cones are concentrated in the fovea centralis. Very few rods are there, whereas the rods you're going to see all around the rest of the retina. The three colors that we've got are red, green, and blue, and then as those signals are sent to the brain is going to reconfigure any sort of color in between. You can see the different shapes of rods versus cones in this image here. Rods and cones are going to send signals through a series of neurons, and then we are going to end up with a neuron that sends axons from their spot on the retina. They're all going to funnel to a certain spot where they all exit together as a bundle, and when they exit together as a bundle, they're coming out as the optic nerve. Because what did we say nerves were? Nerves were bundles of axons in the peripheral nervous system. If these are bundles of axons all converging, kind of like a funnel, and all leaving at the same spot, can there be rods and cones in that location? No. So going back to this page where we say there are no rods or cones on the optic disc, that area where all those axons are all leaving, there's no room for rods and cones. So any sort of light that bounces to that spot is your blind spot. And I showed you the blind spot with the rectangle with the dot and the plus sign before. As my honors friends, I expect you to look more deeply at the retina itself. Zooming in on the retina, you can see that there are multiple layers of cells that are involved. What we actually seems odd, but what we see at the surface when we're looking at the inside of the eye, like the model, you say, oh, this peachy orangey layer is the retina. Well, what you're touching is actually this collection of axons that are traveling across. And then as you go deeper into the retina from that perspective, you've got this ganglionic layer, this inner plexiform layer, and the bipolar layer, the outer plexiform layer, the photoreceptor layer, the pigment cell layer. All of this has to do with the nervous tunic, the retina. And then you've got the choroid, which is the part of the vascular tunic that's superficial to it. So the light passes through all of these layers to be absorbed by the pigment layer, because that's what pigment does, is it absorbs light. And then it's going to act on these rods and cones. Here's what I expect of you. You should be able to, with an image like this, you should be able to tell me the photoreceptor layer, the bipolar layer, and the ganglion layer. The inner and outer plexiforms, that's more for regulation of things, that if you're going to become an optometrist, you can worry about those. But for this level, I need you to be able to label these three layers. The photoreceptor layer, you need to also make sure that you are prepared to tell me that they are comprised of rods and cones. Ooh, that's a terrible underlining. And that cones are for color, and that the rods are going to be for your basic outlining and being able to use in dim light. If you look at the numbers of these cells, where we have 130 million photoreceptors, rods and cones in our retina, look what happens to between that and the bipolar cells. It drops down to 6 million, which means that these rods and cones have to share bipolar cells. And then when we look down at the ganglion cells, we only have 1 million of those. So what we're able to absorb as light photons, we're only able to converge and send out to the brain in a minimum number. What I want you to notice, this is significant, I think, is over here, these ratios. Our ganglion cells are the ones that are sending the information to the brain as the optic nerve. For every one cone, it's going to communicate with one ganglion cell. And I'm sorry that I can't tell you how many cones there are versus how many rods, but I can tell you that there's a 1-to-1 ratio. For every cone that we have, specifically in that macula lutea, one ganglion cell is going to take it. So that means all the information our cone has is going to the brain. Whereas there's a 1,000-to-1 ratio between our rods and our ganglion cells, which is why when I say that the rods are used in dim light and you get these fuzzy images, well, you've got all this information, but it's all being shared on one ganglion cell. And so the brain really can't do a whole lot with that. How do these photoreceptors function? Oh my gosh, it bugs the crap out of me because it seems counterintuitive. When it's dark, so when there's no light, these rods and cones are releasing a neurotransmitter that inhibits the bipolar cells, meaning it turns them off. So if the bipolars are turned off, they can't send an action potential to the ganglion cells. And if the ganglion cells aren't getting a signal, then they can't send a signal to the brain. And if there's no signal to the brain, then the brain says, well, must be dark. So then flip it and say, okay, now there's light. Light comes in and it turns off the rods and cones. So if it turns off the rods and cones, can it secrete its inhibitory neurotransmitter? No, because it turned off. Well, if you turned off the stopper, turn off the inhibitor, bipolars then turn on. And they send a signal to the ganglion cells, which can generate an action potential to send it off to the brain. It bothers me if the rods and cones are on in the dark and they get turned off in the light. And remember my phrase before of the lack of signal is a signal in and of itself? If you turn off the rods and cones, that means everything else can be turned on. Messed up, but that's how it works. Neuronal pathways. Well, what happens once you've got this signal generated? Well, the eyeball is a sphere. And so when the information from your field of view, you've got everything from the right side of your field of view is going to go to these sides, the left sides, if you will, of your eyeballs. And then all the information coming into your eyes from the left side of your field of view is going to the right sides of your eyeballs. Well, we need to join that information together. So when you look here, you've got this crisscross, this optic chiasm, and you see how everything from the medial side, so from the nose side of things, are going to crisscross. So then everything from the left side of my world is going to end up going to the right side of my brain. Everything on the right side of my world will end up going to the left side of my brain. From there, where do these signals go? Well, some of the signals are going to branch off and go to the superior colliculi. Remember, those were the butt cheeks on the midbrain? That's for your visual reflexes. And then other branches are going to go to the thalamus. Remember on that summary chart, I said, hey, the olfactory nerves were the only ones that didn't synapse in the thalamus, but everything else did. Well, here's our synapse in the thalamus before we send all of our information back to the occipital lobe. Isn't that strange that the information we get from the front of our face is going to be processed in the back of our brain? You would think it'd be closer, but it's not. It's directly opposite. So for people who end up with trauma to the back of the head, what are some things that they ask about? The clinicians are going to be like, hey, How many fingers do you see? Because they could have double vision, or they can have blurry vision, or they could have dark spots because the gray matter of the occipital lobe is kind of messed up and can't process the signals that are being sent to the brain. Something I found very interesting was that my father, with his brain tumor, the brain tumor was on the right side of his brain, and it affected the left side of his body. Well, not only did it affect the motor portion of his body, but he also, when they dug the tumor out a second time, somehow these fibers that were running through were affected. And he started to just not notice things on the left side of his body either. So I could place something just outside of his line of focus, like out of his periphery from his right eye. He didn't know it was there because somehow it was affected, those signals going back to the brain. It was so strange. So here is your processing of your vision, information about where these neurons from these ganglion cells are going. In that synapse of the thalamus, it's saying that those neurons are going to go to the occipital lobe. Okay, fine. And there's two kinds of cells in the occipital lobe. The simple cells, they're the ones that are going to get the information and say, there's your object. Here are all the details. Let's put all those details together to give you how complex the thing is that you're looking at. The complex cells, on the other hand, they're going to take the information from the simple cells, and they're going to be the ones that keep the shape, the integrity of your object if it's moving. So if an object is moving and you're tracking it, the complex cells are the ones that make sure that your shape stays intact as you're following it. The superior colliculi, the synapsing there, that is going to be, we move our eyes towards a light, number one, to be able to see an object. Number two, there's a very close relationship between the superior colliculi for visual reflexes and your inferior colliculi for the hearing. When you have a sound that comes to your inferior colliculus, what do you usually do? When you hear a sound, your eyes move to find where that sound came from. Same thing, I mean, that's why, because those colliculi are right on top of each other, and they're able to synapse with each other. So if the hearing comes first, your eyes will then respond. If you see something and you want to focus more on the sound that's coming from where you're looking, you're able to communicate with your ears to kind of dampen the excess noise, and you can focus on the area that you want to listen more intently. This next portion is going to be about color blindness. Cones are for color, but what about the people who can't see color?
Hey there, welcome to Life Noggin. I'm Blocko.
And I'm Sabrina from the YouTube channel Nerdy and Quirky.
And we're here to teach you a little bit about color blindness. There's nothing I love more than seeing the sunset at the end of the day, but for some people these sunsets might not be so spectacular.
See, in the retina of our eyes, we have light-detecting cells called cones. These cones contain 3 different types of photopigments, which are sensitive to different wavelengths of visible light. And while the wavelengths detected by each cone tend to overlap, it is easy for us to categorize the cones as being red, green, or blue.
If you have normal color vision, your red, green, and blue cones are all working properly, and you'd be called a trichromat. However, some people are dichromats, meaning that they only have 2 types of cones instead of 3.
This typically affects about 2% of the male population, who are either missing their red or green cones. These people can't distinguish between red, orange, yellow, and green, which definitely makes those sunsets a lot less interesting.
A more common form of color blindness is called anomalous trichromacy, which affects about 6% of males. And this is where the famous red-green color blindness belongs. People with this have all 3 types of cones, but some of the wavelengths covered by the green or red cones are shifted.
And if the wavelengths from the green cones are shifted, you can think of the person as being green weak. It'll be hard for them to notice small differences between red and green, but it's not as severe as dichromacy.
And since the red and green pigment genes are located on the X chromosome, men are much more likely to have have color blindness. After all, women have two X chromosomes, so both would have to be defective for them to be colorblind.
So if you are a man watching this, take a look at this picture. If you can see a 2 in the center, you probably have normal color vision. But if not, you may want to go to the eye doctor to get officially tested.
Contrary to popular belief, color blindness doesn't mean that you see the world in shades of gray. That does happen sometimes, but it's much less common. Anyway, so tell me in the comments, if you had to describe the color green to someone who's colorblind, what would you say? And are you colorblind? Do you have any experiences you'd like to share? Let us know in the comment section below, and if not, tell us what you would like us to talk about next.
Huh, that's interesting. I don't know how I would describe green to somebody. Here's an image. What do you see? Do you see a 21? Or do you see a 74? If you see a 74, you're normal. Good job. If you saw a 21, you might have some red-green color blindness. How about here? Anybody see a 4? How about a 2? 42? 42 is normal. What if you can only see a 2? Well, you consider red color blindness. And if you can only see the 4, you actually have a green color blindness. Which is strange to me, given that the 4 to me has reds, whereas the 2 seems to have pinks. So, I don't know. That's just me. Now let's finish off with people who have vision clarity issues. A normal eye, emetropia, is where when the light comes through and goes through the lens, it comes to a point directly on the macula lutea, the fovea centralis. Spot on. And that's perfect vision. A myopic eye is where the lens is shaped strangely to where when they converge, it comes to a point somewhere in the vitreous humor. So if they give you a concave lens, it's going to help stretch that light out so that it converges back on the fovea centralis of the macula lutea where it belongs. This was my problem, and I have a little bit of that still. Now the hyperopic eye, farsightedness, that is when if you were going to follow the light rays through the eyeball, they would not converge until past the fovea centralis. Well, since it never gets to the point where it's focused, you get the signals to your brain of it being out of focus. And so they have to use a convex lens, and that's going to converge it closer so that it hits the fovea centralis where it needs to be. Astigmatism. How many of you thought that it was a space stigmatism? I thought that for years. I didn't realize that astigmatism is a condition in and of itself.
Stigmatism, by the editors of allaboutvision.com. For many people, the word astigmatism sounds scary, and they mistakenly think it might be a serious eye condition like glaucoma or macular degeneration. But in fact, astigmatism is simply a very common imperfection in the eye's ability to accurately focus light. Such problems are called refractive errors. There are 3 kinds of refractive errors: myopia, or nearsightedness, which causes blurred distance vision; hyperopia, or farsightedness, which causes blurry near vision, and, if severe enough, can affect intermediate and even distance vision as well. And thirdly, there is astigmatism, which causes objects at all distances to appear blurred or distorted. The severity of the blur is determined by the magnitude of the astigmatism. Most people who have astigmatism have some nearsightedness or farsightedness as well. Like nearsightedness and farsightedness, astigmatism almost always can be corrected with eyeglasses, contact lenses, or refractive surgery such as LASIK. The primary cause of astigmatism is an irregular shape to the clear front surface of the eye, the cornea. One meridian of the cornea is significantly more curved than the meridian 90 degrees away. This gives the cornea more of a football shape than the normal baseball shape that's needed for clear vision. This irregular or astigmatic corneal shape causes light to be focused at several different points within the eye, rather than forming a single, clear focus point on the retina. So vision is blurred or distorted. When astigmatism is corrected, whether by eyeglasses, contact lenses, or vision surgery, light once again focuses directly on the retina, and vision is clear. Astigmatism is easily detected in a comprehensive eye exam. After your exam, your eye care professional can discuss the best method to correct your astigmatism, so you can have clear vision and enjoy life to the fullest. Thanks for watching. For more in-depth information about astigmatism, please visit allaboutvision.com.
And there's just another picture of your two different sightedness. I will post both of these videos later on in the module, along with my LASIK surgery for you to watch. And that will be the end of it. Sorry it was so long.